Hi, I'm really excited to share our work with this group, and mostly I'm excited to see this group growing here. I think there are not enough of us digging out of the genomic side into the functional genomics side, and so I'm going to take it one step further today and not just talk about functional genomics, but dynamic functional genomics. So, how do gene-environment interactions impact our risk for disease? And the question that I've really been asking, for years in the lab then is how do we modify the impact of genetic risk that we're born with? I think in university, we were all taught this linear relationship between genotype and phenotype, genotype in, phenotype out. But the truth is that it's much more complicated than that. There are environments that make our phenotypes more severe. The classic example that I'll often use is post-traumatic stress disorder. You can have all the genetic risk for PTSD on the planet, but if you don't get exposed to a trauma, then you're not going to develop PTSD. And then it goes reciprocally that there are probably environments that make our genetic risk less severe. And can we understand the pathways that are active or repressed in those situations to minimize risk and use that to prevent risk in all of us? The goal here is really to achieve our genotypic maximum, the best possible outcome with the genetic risk factors we are born with, independent of gene therapy. And so that's what the lab has been asking for the last 5 years or so. How does context modify genetic risk? I have a group looking at how the rest of our genetic background modifies the impact of an additional risk factor, how risk factors have different effects across development and into aging. Why do Alzheimer's risk factors only seem to matter when we're old? How risk factors have different effects across different cell types. The impact of sex. I think this is a really interesting question. Why are some diseases more likely in males and others more likely in females, particularly when they're not driven by factors on the sex chromosomes? And then I think more broadly, how does the environment, and these are classical environments in a dish, the addition of stress hormones, inflammatory cytokines, and neuron injury, metabolic changes, drugs, and toxins, how do those impact the impact of risk factors? Oh, my slide. Oh, okay, now I'm going to start with gene environment. So, briefly, many of us will be exposed to a trauma over the course of our lifetime that would be sufficient in magnitude that it could be associated with a diagnosis of PTSD. But only 10 to 20% of people who are exposed to trauma go on to develop PTSD. So what determines how we respond to trauma? Well, the largest determinant is actually previous trauma exposure. So those people who've had more previous trauma exposures are more likely on the next trauma to go on to develop it. But we wanted to know as well if there were genetic predictors of people who are more likely to develop PTSD after trauma exposures. So, really asking, do our genetics determine how we respond to traumatic stress? And so to ask this question, I was really lucky to work with Rachel Yehuda at the Bronx VA. Rachel had recruited 39 combat-exposed veterans and had blood samples from which we reprogrammed stem cells in collaboration with the New York Stem Cell Foundation. From those stem cells of 39 donors, we made induced glutamatergic neurons and then did RNA-seq at baseline with low-dose cortisol and with high-dose cortisol. And so what you're seeing here is that with low-dose stress hormone, a few genes change expression, but genome-wide, if you stress the neurons more, there's a lot more changes. This makes a lot of intuitive sense. The more we stress neurons, the more they respond. But if you break this cohort down into those neurons from individuals with and without PTSD, it actually shifts the way the data looks a lot, right? Because now we see a stronger PTSD signal at low dose and less of a PTSD signal at high dose. And what I think this data is telling us is that everyone's neurons respond to high dose cortisol, but only those neurons from those individuals who had PTSD after combat exposure, they responded at low dose. So their neurons were hyper responsive to cortisol, at least in the dish. So we asked what types of genes were hyper responsive to stress in those individuals who had PTSD. And in fact, those genes that were hyperresponsive were broadly linked to many psychiatric disorders. We did see enrichments for PTSD gene sets, but they were not specific to PTSD. We also saw enrichments for neurodevelopmental disorders, schizophrenia, epilepsy, and autism. And so broadly, we concluded that neurons from PTSD cases showed hyperresponsive to stress, grossly affecting many psychiatric risk genes. And so how did this happen? What is the mechanism for this dysregulation following stress? Well, we've talked a lot today about genetic variants that regulate expression, and I'll use the term EQTL for these variants. They're typically found in promoters or enhancers, although they can be, of course, in 3' UTRs. In this particular toy example, somebody who inherited the G allele would have less expression than somebody who inherited the A allele. But now I want to introduce this idea of a stress-interactive EQTL. So this would be a variant that has a different effect on gene expression in this given tissue, dependent upon your stress or trauma exposure. So in this particular example, at baseline without stress, again, that G allele will have less expression, the A allele will have more. But as you show accumulating stress or trauma exposure, in this particular toy example, we're seeing a flip in the relationship, with now the AA having less, the GG having more. And what's important to note is you would miss this in any EQTL analysis in humans unless you had annotated trauma exposure as well. So we wanted to go looking for it. We were incredibly lucky to get to work with Matt Girgenti here because he had a postmortem, the only postmortem cohort that I think was suitable for this examination. 304 postmortem brains, RNA-seq, already genotyped with annotated trauma exposure. So these were, again, combat-exposed veterans now at the New Haven VA. And Matt gave us all of this data really generously. And so Karina went in and asked, could she find variants that showed differential regulatory activity in those brains that had more trauma exposure? And then to validate it, because there was no second cohort, she actually went back to our stem cell models. And here by now we had glutamatergic and GABAergic neurons from those 39 donors with and without cortisol. And so by intersecting this data, Karina was able to show that trauma and stress interactive variants were enriched for transcription factor binding sites. Some of those transcription factor binding sites are, the usual suspects. NF-kappa B, one of the best inflammatory mediating transcription factors. Really reassuringly, glucocorticoid receptor, the receptor for the stress hormone that we were adding to the neurons. And then one here, YY1, that had come up in the previous study, and we decided to validate. So this is just a generic RNA-seq, 2 controls, glutamatergic neurons. If you stress them, some genes go up, some genes go down in red. Now in gray, we're adding the effect if you add a short hairpin to YY1. And so if you block YY1, you blunt the neuronal response to stress. Genes that were supposed to be way up went less up, genes that were supposed to be down were less down. And so you're blocking the effect. At this point, Karina graduated. She's gone back to her MD PhD studies. In fact, she graduated from her MD a couple of months ago, and we're hoping to get this revision back out and updated before the end of the summer. But in parallel, Kayla was another graduate student, also co-mentored with Laura Huckins. Whereas Karina was really interested in the effect of stress in veterans, Kayla is really interested in the effect of stress and inflammation on fetuses in utero. So how do things that happen to the mother while pregnant impact the outcome of offspring years and decades later? We know from epidemiological experiences that women who experience infection, famine, war, and trauma in second trimester are more likely to have offspring with autism and schizophrenia. And so we wanted to ask how inflammatory cytokines might mediate this. We're going to use, we use 3 stressors in a dish. We chose IL-6 because it is thought to be one of the mediators of maternal inflammation. It accumulates in the placenta and is thought to cross the blood-brain barrier. We use interferon because it's a fetal cytokine. And so we wanted to see how the fetal cytokines might impact gene expression in the brain. And then our high-dose cortisol is a benchmark. The first thing that I want to point out is that the neurons responded differently to each of these stressors. It's not that neurons had 2 states, resting and reactive. We saw distinct genes being up and down regulated across each of these cues. And so the question here is very similar. Do our genetics predict how we respond to prenatal stress? But I'm going to be upfront with you. What we did not have is a collection of stem cells from offspring born of mothers with known exposures 20 years prior. Like that would be an amazing cohort, but I don't have it. And so we had to do this experiment more synthetically. Kayla generated a massively parallel reporter assay. So this is an assay that had, in this case, 10,000 variants pulled from 220 GWAS loci associated with schizophrenia, bipolar, autism, ADHD, OCD, depression, anorexia, PTSD, neuroticism, and Alzheimer's disease. This library was designed around 2020, so the GWAS are a bit outdated, and unfortunately, as it goes, we made the choice at that time to prioritize the variants based on EQTL co-localization, which I wouldn't do again, and I'm happy to discuss the implications of, but nonetheless, we had a very large library, and we put them into neurons, and then we treated the neurons with inflammatory cytokines, and we asked how the regulatory activity of all these GWAS loci varied in neurons by context. So some of the EQTLs were stable, which is to say they had the same regulatory effect across context. And here's an example here, while the alternative allele, whether at baseline or IL-6 or interferon, was always less. Here's an example of a dynamic effect. In this case, we have a SNP that only downregulates expression in the context of interferon and not at baseline or with IL-6. And then Kayla went into the library genome-wide and asked how our risk variants showed regulatory activity in human neurons. And so at baseline, it won't surprise you to see that we saw enrichments for things like schizophrenia and bipolar and depression. These were exactly the variants that were in the library. What's more interesting is to see how these enrichments change with exposures. So here, for example, is schizophrenia at baseline, and it's significant, but with different extents across all the contexts. More interesting, I think, is this subset of schizophrenia risk variants that are associated with pleiotropy for metabolic syndromes, which only were significant in treatment with IL-6. And then more broadly, some of these schizophrenia enrichments are also linked to pleiotropy that came up with either with IL-6 or with interferon. And over, we saw this same pattern where the pleiotropic variants were the ones that were dynamic and the cleaner disease variants were there at baseline. It was really interesting. And I think the take home of this data analysis, which is also ongoing, is that the weather matters, right? It's not just about sunny weather or storms, but the type of storm. Your genetic risk might be more predisposed to blizzards, and your genetic risk might be more predisposed to hurricanes, right? And so the type of outcome you show will really reflect both your genetics and the unique environments that you're exposed to over your life. The second half of the talk is not about gene-environment interactions, but more about gene-cell type interactions and how these genes converge across complex genetic diseases. So we're gonna, here for a moment, stay in our common variant effects. This was a prioritization of genes done from the PGC3, where we co-localized EQTLs in the brain with GWAS SNPs, and also used PredictScan to prioritize what we thought were putative causal genes being regulated by schizophrenia GWAS loci. And so that prioritization at the time, around 2020, 2021, brought up 25 top genes regulated by schizophrenia GWAS. And we pulled from those the top 5 genes with synaptic function, the top 5 genes with epigenetic or regulatory function, and the top 5 genes that were not synaptic, not regulatory, and that had no apparent relationship to each other. And what we wanted to ask was what types of genes are downstream of these genes if we perturb them in the schizophrenia relevant direction. So for those genes that the GWAS and EQTLs predicted were up in schizophrenia, we upregulated them, and for those that were down, we perturbed them down. And then we asked downstream what genes were changed. So within the 5 synaptic genes, we were looking for downstream genes that were up 5 out of 5 times or down 5 out of 5 times. Same within the regulatory genes, differentially expressed genes that were consistently up or down across all perturbations. So for the 5 synaptic genes, we found about 1,000 convergent gene targets, genes that were changed in the same direction. And surprisingly, with the regulatory genes, we also found about 1,000 convergent targets. But with the multifunction genes, we only found 89. And so we saw less convergence for these multifunction genes. I also want to add that the convergent networks were unique between the synaptic genes where we saw and the regulatory genes. So unique compositions of networks. They're not the same convergent genes downstream of both groups. We wanted to ask if we could predict drug targets Not the schizophrenia risk genes, but in the downstream convergent targets. Do we have to reverse schizophrenia risk genes at their source, or can we reverse them downstream? And so this was done by using the LINKS data set, looking for drugs that reverse schizophrenia network signatures. One of our top hits was a mitochondrial modulator, itoxamir. And here I'm showing the RNA-seq effects of CALN1 in blue and CALN1 with itoxamir, so a reversal of the perturbation direction. Same drug, different schizophrenia risk gene, perturbation of TMEM219 in green, and a reversal of the transcriptional signature in yellow. And so again, we're not targeting schizophrenia risk genes, we're targeting convergent downstream effects. We did the same analysis, but with the rare genes linked to autism. So this is pulling from the Satterstrom data set in Cell. Here they have 102 autism genes. Now, the schizophrenia risk genes we inherit in combination, but in autism, each individual inherits a different risk gene. We pulled the top 30 autism genes that were chromatin modifiers. Since we're going to do a transcriptomic assay, we thought we'd give ourselves a fighting chance, focus on gene regulators. We did this CRISPR screen in neural progenitor cells, in immature and mature glutamatergic neurons and mature GABAergic neurons. And again, we found convergent effects that were more similar within cell type than without cell type. And so as a geneticist or as a neuroscientist, many of us seem to have our favorite gene. My favorite gene is Nerexin-1. But what you can see is that I would learn a lot more by knocking out ARID1B in iGlut to learn about Nerexin-1 than I would about knocking out Nerexin-1 and IGABAs, right? You can learn a lot by knocking out different autism genes in the same cell type. The strength of convergence was actually greatest in glutamatergic neurons. But again, it wasn't that the same convergent networks got stronger with differentiation. We had unique sets of convergent genes and convergent networks in each cell type, with convergent networks in glutamatergic neurons being really enriched for synaptic function. The reviewers did what reviewers do. They said, that was great that you looked at 30 autism genes, but there was 102 in Satterstrom. What happens if you go perturb all of them? Now, we couldn't do that, I'm going to be honest, and instead we worked with Mark Gerstein to apply the AI models that he generated from his 388 single-cell brain RNA-seqs. And the AI model went back and tested not just the 30 genes we'd already done, but all 102 of them. And what he showed first was that he could reproduce that convergent signature that we saw stronger in glutamatergic neurons and weaker in GABA. He saw the same thing, stronger predicted convergence in GLUT, less in GABA. And then almost reassuringly, he showed that if we had done the experiment in synaptic genes, we probably would have seen a stronger convergence there. And that actually tracks with our schizophrenia report where we showed stronger convergence with the more synaptic genes in a gene set. Again, we can see his AI predicted the gene ontology of the convergent genes by risk set, whether they were synaptic or epigenetic or linked to developmental delay or autism. In all cases, we saw enrichments for synaptic and epigenetic, but it wasn't that the epigenetic risk genes target on the synaptic. Synaptic genes converge on synaptic and epigenetic. Epigenetic genes converge on synaptic to epigenetic. It was less hierarchical than I had imagined going in. But unexpectedly, also a mitochondrial function, which is not something on my radar, something our lab thinks about a lot. We actually spent 6 months trying to validate this. We could see in our own empirical data that same enrichment for mitochondrial function when we looked for it. We did 2 CRISPR screens asking if our 30 autism genes impacted mitochondrial function, here using an mitochondrial membrane potential dye as a readout, and almost all of them did. We also confirmed differences in mitochondrial morphology and by the gold standard assay, Seahorse. And interestingly here, the Seahorse assay showed a deficit in Nerexin-1, which I already told you was my favorite gene. And what I was really surprised by is that I learned about this deficit in Nerexin-1 mutant neurons by studying all of the autism genes, right? Not by studying Nerexin-1. I learned this as somebody who has studied patient-derived Nerexin-1 knockouts for 15 years. I learned this by perturbing 30 autism genes. I know I'm running short on time, but I do want to quickly highlight Nerexin-1 because it's such a really cool gene and such a good example of why we need to do functional genomics, and not just in mouse cells, but in human cells. So Nerexin-1 is highly penetrant, also pleiotropic. It's linked to both autism and schizophrenia, as well as epilepsy and OCD. It's highly alternatively spliced. This is data from the mouse brain, over 350 Nerexin-1 isoforms. And the heterozygous phenotype is human-specific. This is data from Tom Sudoff. So when you compare the exact same mutation in neurons made the exact same way from mice and humans, you only see synaptic deficits in the human neurons. Likewise, you don't see behavioral deficits in mice until you have a homozygous knockout. Together with Gong Fang and work led by a former PhD student, Aaron Flaherty, and a former postdoc, Xu Zhu, we looked at the splicing in human brain and human stem cell-derived neurons. Like others had shown in mouse, we also saw complex alternative splicing repertoires. And reassuringly, we showed that the most abundantly expressed splice isoforms we detected in both human and stem cell-derived neurons. So we felt really good now trying to model patient mutations in stem cell models. So here we've got 4 patients. I should add that mutations in orexin-1 are non-recurrent, different boundaries in different individuals. And we are really fortunate to actually have biological replicates of the same non-recurrent mutation, because what we're calling our 5' deletions was a variant passed from the mother to her son. Both were recruited with a bipolar with psychosis, and 2 monozygotic twin pairs with mutations in the 2nd, 3rd, and 4th from last exons, both recruited with adolescent onset psychosis. So we wanted to know to what extent phenotypes in these neurons in the dish reflected perturbations in few or many neurexin-1 isoforms. This is all published, so I'll go pretty fast. What we're able to show is gross dysregulation of neurexin-1 splicing in the 3' mutant neurons. Half the isoforms were significantly decreased. Another third we didn't even detect. I think this was a limitation of sequencing depth. So massive dysregulation of wild-type isoforms in patient-derived neurons. And then really unexpectedly, 31 unique mutant isoforms caused by splicing around that red deletion from exon 20 to exon 24. And what you can see in purple is that these mutant isoforms were among the most abundantly expressed isoforms in patient neurons. So they're really, really highly expressing these mutant isoforms. We wanted to know if these isoforms were expressed, so we cloned them. We confirmed that by Western blot that we could see tagged versions of them. Then we overexpressed them. Overexpression of a single mutant isoform was sufficient to impair activity in control neurons, phenotype being the phenotype we saw in the patient. So we think that might be a gain-of-function mutation here. In those 5' mutations who we do not think make mutant isoforms, we could rescue phenotypes by overexpressing a wild-type isoform. But in the 3' mutations who do make mutant isoforms, we could not rescue by overexpressing a wild-type isoform. So we think there's 2 types of mechanisms at play. In a subset of patients, pure loss of function by loss of neurexin-1 dose, and in a smaller subset, gain-of-function mutations through a mutant isoform activity. Now, was I so lucky that if I made stem cells from 4 people, 2 of them happened to have the only gain-of-function mutations known in genetics? And the answer is no, I don't think I'm that lucky. We went into the PGC3 data set at the time and predicted that about 1 in 5 neurexin-1 carriers might have mutant isoforms that are translatable. There's exactly 1 neurexin-1 postmortem brain tissue that I'm aware of. It's at the Lieber in Baltimore. They gave us some RNA from that brain. It is predicted to have a deletion in exons, I think 10 to 14. We found mutant isoforms in that brain as well. So in 2 of the 4 IPS lines we've tested and 1 of the 1 brains we've tested, we've actually found evidence of mutant isoforms. Aaron graduated, and with my last data slide, Michael carried this analysis forward. Now he's looking in pure glutamatergic neurons, pure GABAergic neurons, same 4 genotypes. And what he's showing here is reduced excitability, as Aaron reported, in glutamatergic neurons, but in pure GABAergic neurons, hyperexcitability in immature neurons. So the same deletions having very different synaptic effects in different cell types. He actually reproduced this by electrophysiology, patch clamp, the gold standard, reduced EPSEs and increased iPSEs in patient neurons. And he demonstrated, again, the loss of function by knocking down wild-type isoforms in control neurons and knocking down mutant isoforms in those patient neurons. So again, I think really demonstrating these are both loss and gain-of-function mutations with distinct phenotypes in different cell types. Now, Michael is so translationally oriented that he wanted to show that it was possible to rescue these phenotypes in post-mitotic neurons. So how to rescue this loss of function? We had already shown that you could overexpress a wild-type isoform, but they're really big. This is not going to work for gene therapy. Could we re-express it from the endogenous allele in the wild-type side? Well, 2 pieces of evidence made us think it was possible. First of all, the estradiol receptor by ChIP-seq binds the promoter of Nerexin-1, so maybe that would activate expression. And two, you can actually rescue phenotypes in Nerexin-1 NPCs by adding estradiol. So we added estradiol. We were able to show that increased Nerexin-1 expression in glutamatergic neurons, but interestingly not in GABAergic neurons. And it completely rescued the synaptic deficit by adding estradiol to these 5' neurons. For the loss of function, Michael designed ASOs to target Nerexin-1 mutant isoforms in the I-glutes and the I-GABAs and showed a rescue in both. He wasn't able to patch from these ASO-treated neurons, but I'll remind you of our short hairpin data showing that if we knock down mutant isoforms, we can achieve a rescue. So the idea I want to close with is that sex hormones might actually modify the impact of genetic risk, and this might, be informative and reflective of the fact that many brain diseases do have strong biases by sex. Autism being more common in males, Alzheimer's, depression, and anxiety being more common in females. The idea to close with is that gene-environment interactions inform genetic resilience, that there are genetic superheroes who do not live with the phenotypes that their genetics suggest they should be. And so if we can understand the mechanisms of this protectiveness, perhaps we can find drugs that will help more of us. And with that, these are the folks who did a lot of the work I talked about today, and I'm happy to take questions if there's time.